I'm delighted to have with us Professor Peter Hegemann. He's from the Department of Biophysics at the Humboldt University of Berlin, Germany. Professor Hegemann studied chemistry in Münster and Munich and earned his PhD in 1984 in Munich, working on the structure and function of Channelrhodopsins. You'll hear more about this. It's a light-driven chloride pump from halophilic archaea. As a postdoc, he worked in the research groups of Dieter Osterhag, the famous Dieter Osterhag, and with Kenneth Foster in Syracuse University. In 1986, he started a research group in the Department of Membrane Biochemistry at the Max Planck Institute in Frankfurt, and became a professor at the University of Rosenburg, and was appointed full professor at the Humboldt University in Berlin in 2004. Professor Hegemann was the discoverer of Channelrhodopsin, or Channelrhodopsins, a family of directly light-gated ion channels. This discovery has opened a new field of optogenetics, that is, the manipulation of the activity of neurons and other cells with light. Since the original discovery of Channelrhodopsins, Professor Hegemann and his group have improved its properties by molecular engineering, resulting in a wide variety of designer opsins, which are faster, more sensitive, responsive to different colors of light, or conduct different ions compared to natural Channelrhodopsins. These molecular tools have enabled neuroscientists around the world to stimulate genetically defined populations of neurons non-invasively and with great precision. Professor Hegemann won numerous awards and honors, for which I'll mention just a few. He is a member of the German Academy of Sciences, a member of EMBO, and he is a winner of the Technion-Harvey Prize. So, Peter.
So, ladies and gentlemen, dear friends, I think this is a fun part of the Harvey Prize because I'm back in biology, and I feel much more familiar in biology than I feel in the neuroscience, because I don't understand anything, and I'm a neuroscience professor now, but I never did a neuroscience experiments myself. So, I'd like to explain a little bit about the history of the Channelrhodopsins before I go into more recent studies. And my work started actually after I finished my PhD with organisms like this, green algae. This is Volvox and several other organisms. And now we work on the details like this and the mechanism to understand the transport function and so on. And algae are also nice because they smell nice, and especially in Japan, they taste nice, and there are several reasons why algae work is pleasant. And also, if you look at them, not only in the light, but also the fluorescence is very, very appealing. So the algae we used over the years is Chlamydomonas, most of all. It's a unicellular alga with an eye spot, and this is Volvox, many cells at the surface, eyes looking all the same directions. And this is Euglena, also an alga that has been studied more than 100 years. And this is Acetabularia, benthic algae of the Mediterranean Sea. And during reproduction, it also produces single cells that are motile and have eye spots. And I'd like to start with this publication. And you see the business is not as new as one might think. So this is an interesting publication from Faminsin, Professor Faminsin of St. Petersburg, and he described the behavior of Chlamydomonas very nicely. And as a Russian professor, written in German, published in a French journal, which is kind of surprising. And this ancient German he used is very funny to read, and he describes it very, very detailed. And this is experiment. Unfortunately, there are no figures in it, but Simon Kelterborn in my group has made a drawing. So first of all, it's interesting that he only could use sunlight because electricity was not available at that time. So if you consider your experiment without having electricity, would be more difficult. And so he used open dishes without any edges, and after illumination, the algae accumulate in the on this plate at the border of the covered side of the plate. And at low light, so late afternoon, algae accumulate at the front part. So this called dish test has been used over many years. So this is 1866, and almost 100 years, the progress about the photoreceptor of this alga was practically zero. So many German groups worked on photoreceptors, on the isolation of the photoreceptors in many different algae, including Chlamydomonas and Volvox, and none of them was able to make a significant contribution. And the Euglena receptor, for example, was identified by Watanabe, Masakatsu Watanabe in Tokyo, after many German botanists failed. So we work with Chlamydomonas most of the time, and we know the beating behavior pretty well. And this is controlled by the eye spot, which is integrated into the large chloroplast. And recently, Ben Engel in Wolfgang Baumeister's group made very nice figures of the eye spot with cell tomography. And I think tomography is the method of the future, and it will revolutionize cell biology. And what Wolfgang Baumeister said to me about 4 weeks ago, in the next 10 years it will be possible, very likely, that you can determine three-dimensional structures from just tomography recordings from single cells. So also the beating can be certainly studied now in every detail. And this recording has been done by Kirsti Wen in Raimund Goldstein's lab in Cambridge. So we have very detailed information of the beating behavior. I'd like to mention this experiment. So more than 100 years, Ken Foster, a physicist from Syracuse University, published a paper together with Koji the Magic, was a chemist from Columbia University, where they supplemented Chlamydomonas cells that are blind, so white cells that do not produce carotenoids or retinol, and they use this simple dish test. This is a figure from my lab, and so a test that was more than 100 years old, and proposed that the photoreceptor for this organism is a rhodopsin, and they proposed that this is an rhodopsin which is similar to our rhodopsins in the eye, so an animal type rhodopsin. Okay, so just using this dish test, they made a very important proposal. And I spent a year in Ken's lab to study this photoreceptor. We didn't get very far. I shared a cold room with Benny Horwitz. We worked together there, and both of us were struggling for photoreceptors. And that was not so easy in this situation. So then we went back to the behavior. This is Euglena, for example. If you switch on the light, you see that it turns into a tumbling, and we quantified the reactions. This doesn't work, well, as usual. So you should see the Chlamydomonas. I don't know why it doesn't work with a projector on. And you can monitor individual traces, monitor the stopping and everything. This is a pity that it didn't work. And then we established electrophysiology for Chlamydomonas, and this is seen here. This is probably the most important experiment we ever made. And we used the suction pipette technique and sucked the alga into that large pipettes. And an important observation was, by the way, done by my second graduate student, Hartmann Hartz. And he showed that flash simulation generated a fast photoreceptor count followed by a flagella count, which means ion influx into the eye and ion influx into the flagella as well. So we characterized this photocounts in detail, and the action spectrum was a rhodopsin-shaped spectrum with a maximum close to 500, and we proposed that these photoreceptor counts are mediated by the rhodopsin. And we studied these photoreceptor counts, or these, over the years. And one interesting experiment is shown here. We monitored simultaneously the flagella beating with the photocounts that are evoked by a short flash. And what you see is when the photoreceptor count and the flagella count occur, this beating pattern disappears. And the reason is that the monitoring spot is here at the side, and they switch from forward swimming to backward swimming. And this is a basic for later application, because it's exactly what the neuroscientists want to do. They want to produce action potentials based on primary photoreceptor counts. So what the algae is doing since millions of years. At very low light intensity, no action potentials occur, and then you get a small beating change, which makes a directional change of the algae, but no action potential. So this was a combination of electrical recording with optical trapping, which was quite successful. And over the years, we improved the system by directly recording the photoreceptor count by using a real patch pipette, and which means photoreceptor counts are inverted because the count flows from the pipette into the alga. But the advantage of this was that you get high resolution, which means a photoreceptor count occurs or begins to rise with a delay of less than 30 microseconds. And we made several conclusions out of it. The first one was the rhodopsin and the ion channels are directly coupled. The second one was they form a light-gated ion channel. And the third was a channel conduct proton and calcium. And that was a very difficult time because no one else believed that the rhodopsin and the ion channel could be one unit. And we were heavily criticized for all three statements. The other thing is that the ion conductance that we calculated is in the range of only 100 femtosiemens or below, which was not considered as a real channel. And that was a problem, and still is a problem up to now. So the problem was solved several years later in a collaboration with Georg Nagel. And my graduate student, Sunil Kataria, identified the photoreceptor proteins in a Katsuza database from Japan. After we spent several years on the wrong rhodopsin, I skipped this part of the story, which was very time-consuming, and we invested 10 years in the wrong protein. But finally, we got the right one from the Katsuza library, and they looked like this: 7 transmembrane helix proteins with large C-terminal ends, and we injected this we made first RNA and injected the RNA into frog oocytes and used a 2-electrode voltage clamp system. And what you can see is this produces photoreceptor counts, which are very similar to the photoreceptor counts previously observed in the alga. And if you illuminate it longer, you get stationary counts as well. And these inactivate to a stationary count, which is a problem for neuroscience application, and come back to that point later. An important application was also the expression in human embryo kidney cells, because these were the first animal cells. And we named these proteins Channelrhodopsins because, as expected before, they unify the photosensor with the ion channel in one protein. And the advantage of this protein is that it is relatively small, because only the transmembrane domain fraction of the protein is necessary for this sensor function and also for the ion channel function. So all the rest of the protein, which is 60%, is not necessary. This is only needed to direct the protein in the eye spot membrane. Okay, so human embryo kidney cells stimulated the neuroscientists also to try the neurons, because they said, and we already proposed this, if it works in kidney cells, why shouldn't it work in other human cells? So let me summarize the biology up to this place. We have the eye spot overlaying plasma membrane, which contains a channelrhodopsin 1 and 2, and it also contains a voltage-gated calcium channel, which we are characterizing at the moment, and the light is concentrated at that membrane due to reflection on this, at this optical system. And we also localized it first in the eye spot, so exactly where it was expected to be. But it's interesting that there's a small fluorescence also at the flagellar basis. And later experiments by Carol Diekmann's lab, and she showed that the protein travels up here, flagellar rootless of the daughter cell, and this allows it to accumulate in the eye spot membranes. Another experiment interesting from a biological perspective is we try to demonstrate this is really the photoreceptor, and for that purpose we applied an antisense approach, which means you can express antisense RNA or an antisense DNA encoding fragment in Chlamydomonas. It produces antisense RNA, which leads to degradation, and you see the concentration of the channelrhodopsin 1 is high normally in these cells that we use for electrical measurements, and it reduces this to 2%, and the phototaxis, which is shown here in this little dish test again, is almost completely disappeared. Although we introduced antisense approaches for green algae, we never used it later on because the difficulty in Chlamydomonas is that these antisense constructs are rapidly silenced. So after a couple of weeks, you cannot use the strain anymore, which is a big problem. I come back to that point later. So following our expression in kidney cells, the next application was in neurons, and this one was done by Carl Dijsselrood, who gave a presentation here later, and at the same time by Hiromu Yavu in Sendai. And both showed, Carl in hippocampal neurons and the Yavu lab in slices of the mouse brain, that a train of light flashes can produce action potentials very nicely. And that was the beginning of other experiments. The next experiment was done by Hiromu Yavu in chicken embryos and by Alexander Gottschalk in C. elegans. And then experiment followed, certainly in Drosophila and zebrafish, and in mouse brain in living animals. And last not least, and this is a very important contribution by Zuo Pan, he expressed it in secondary cells of the mouse retina and recovered the vision in blind mice. This is a kind of tragedy because Zuo Pan was definitely the person who submitted the first opgenetic paper, but this was rejected twice, so he was relatively late with his publication. But he published the first abstract on a meeting demonstrating that channelrhodopsin can be successfully used in animal cells. Okay, so I don't want to talk about opgenetics more detail because I'm expecting that Carl explained you more about this 2 days ago. Oh, yesterday. Was it yesterday? Yesterday, yeah. So let me summarize. You have to take the DNA from an alga and combine it with the control element of the mouse cells you want to target, and then pack the system into a virus, and then you inject the virus into the mouse brain, and then you replace the needle for a light guide, and then you can study the behavior. And what is needed is that you need a photoreceptor that is relatively small and genetically encodable, and you need promoters that are specific for the cell of interest. If this is not available, you can use Krelog systems. Maybe Carl also explained the systems. And you have to use a chromophore in sufficient amount, and this was a big surprise for myself, that all cells in the brain produce enough retinol, in this case all trans retinol, to complement the opsin into a functional photoreceptor. This was not expected, and none of us expected this because what we learn in the textbooks is that the retinol is only present in the eye in sufficient amount, and this is not the case. Obviously, it is produced in large amount on demand when you suck up all the little traces of retinol in the brain. Okay, so let me summarize the advantage. The advantage of opgenetics compared to traditional methods is that you can activate a single cell out of a large consortium because you can specifically express the opgenetic tools, so light-activated proteins, in one of these neurons and keep the other ones unaffected. And this is certainly also the case for inhibitory proteins like proton pumps or chloride channels or potassium channels or whatever. And this has been used in the past for studying learning and memory, sleep and behavior, locomotion activity, feeding, sexuality, autism, and background for several diseases, as probably Carl explained as well. So I don't want to go into the application. I'd like to introduce you to the biophysics. And very important for the biophysics is certainly the structure. And several groups in the world tried to get the structure. And Osamu Noreki, who collaborated with Carl and myself, solved the structure in 2012 by using a hybrid that we constructed 2 years earlier. It's called C1, C2, because it's a hybrid between the Channelrhodopsin 1 and Channelrhodopsin 2. So maybe I shortly introduce into the structure. This is a chromophore, as everyone understands immediately. Then we have these yellow residues here, and these yellow residues are important for color tuning. So depending on these residues at these positions, we have a variable absorption that may change between 440 nanometers and 600 in these days. We have an access channel. Maybe we start here. So normally this is the extracellular side. Cations are collected by these glutamates and negative charges at the surface. And then they can travel until the central gate, because the central gate is blocked in the dark. And then there's no water between the central gate and the inner gate. And so they cannot go any further unless the gates are open, and then a continuous water pore is formed. And we have an inner gate, and all these amino acids that are listed here were studied in the past by our own group and others. So let me focus today a little bit on the central gate to demonstrate how we work. An interesting feature here are the water molecules. And the water molecules are not visible in the X-ray structure. The X-ray structure only shows the water molecules that are uninteresting. And the reason is that you only can visualize water molecules in a structure if they are not mobile. So for example, water molecules in this place are visible, and water molecules in this place are visible, but those who are important for the conductance are not visible because they can move in and out and change the position in the crystal rapidly. Okay, another method we use for analyzing functional residues is infrared spectroscopy. So what infrared spectroscopy does is it monitors vibrations of any bond in the protein. So these are a couple of thousands, and these it's not possible to analyze. So what we do is we do infrared difference spectroscopy. So we only look at the vibrational changes that occur upon transition from the dark state to the conducting state, for example. So the positive vibrations occur in the conducting state, and the negative ones disappear in the conducting state relative to the dark state. So let me focus on one residue, which is this E90 residue, which is part of the central gate and which is in close connection to the retinal shift base. And we identified this vibration, for example, by mutation. And we see in the mutant, this vibration disappears. So this was assigned as a vibration of the glutamate. And this glutamate deprotonates, for example, upon activation of the protein. This is just one example, and you certainly can do it with many others. And the conclusion was that gating involved large structural changes, including early deprotonation of this glutamate. And so we can, over the years, accumulate information how this gating process evolves. So So let me summarize this part. We know that isomerization or light isomerizes a retinal chromophore around the 13-14 bond. It destroys this bond here between the two residues. This flips down, and then the gate is open. And then we have this situation that the water can move up here into a more or less a water-filled pore. And then later during the photocycle on a microsecond time scale, the inner gate is also open, and then you have a continuous pore which is conductive for protons, sodium, and potassium. Okay, so to describe this also mechanistically, we have to do spectroscopy on different time scales, and this is on one hand the difficulty of the photoreceptor, on the other hand the beauty of the photoreceptor, that you can monitor structural changes over 15 orders of magnitude from femtoseconds to many seconds. And this is an advantage because you can study structural changes which are not accessible for any other biological molecule. So the slow time scale is this one from the first photointermediate, P500. So these numbers just mean the absorption. So it changes from the dark state, which absorbs at 470 to 500. Then the chromophore is deprotonated, loses its protonated shift base, and then you get this blue shifted absorption. And this equilibrates with a conducting state called P520. And then on a 10 millisecond time scale, the channel closes and converts into the P480. So we have a sequence of photointermediates, of structural changes that include movement of the helices, proton transfer reactions between different residues of the protein, in particular the active site around the retinal shift base and along the conducting pathway. And certainly we have rearrangement of the hydrogen bonded network between water and the protein. Now we can ask what happens on this time scale, 50 nanoseconds. And we know from the visual photoreceptors that quite a lot may occur on this time scale as well. So what we have to do is we have to do picosecond spectroscopy that we do with John Kennis in Amsterdam. And what happens is that even on this time scale, quite a lot happens and decisions are made already on a picosecond time scale. So what happens is you excite the protein from the dark state, from the electronic non-excited state to the electronic excited state. You end up in the Frankonden region, and then the protein relaxes to a minimum, and then it even goes down, then nothing happens, so the energy is wasted. Or it goes over a hill, proton rearrangement, and there's a conical dissection, what we call it, and then it reaches the first ground state, which is short-lived on a picosecond time scale, and it reaches on a 4000 nanosecond that photointermediate that we previously observed with a flash photolysis system. Another complicated thing is that depending on the engineering, we might introduce more freedom and more flexibility into the protein, and this can also be disadvantageous. For example, in the C1, C2 Channelrhodopsin that was used for crystallography, the quantum efficiency is only 30% compared to almost 60% of the wild type. And the question is, what is the reason for this? And the reason is that we have several excited states that convert between each other, and only the return of the highest excited state to the first photoproduct is proficient, and the other ones finally go back to the dark state without any productive state later on. And the question is also what occurs, and what might happen is that the retina can isomerize around this bond in both directions, counterclockwise or clockwise. And only one rotation is productive, the other rotation is not productive. Okay, what we also would like to know, we have some indications for that the quantum efficiency is voltage dependent. So depending on the membrane voltage, the efficiency of the proton conversion into a photoproduct is also changed. Okay, another interesting finding, this is more modern spectroscopy now, was a resonance Raman spectroscopy, and I don't want to go into the details. Raman spectroscopy was done at low temperature, and you see then at these 80 Kelvin temperatures that the absorption spectrum is fine structured. So some of the motions are frozen at this low temperature, and what you can monitor is a transition from the all-trans state into a cis-syn state, which is a light-adapted state, or a dark-adapted, light-dark adaptation process in bacteriorhodopsin also involves this isomerization. And what we found is that even at this low temperature, when the protein cannot go through a photocycle, we get light adaptation systems. And the reason is that you can either make a 13 cis 14 isomerization going from here to here. This is a normal photocycle, but also can go from trans-anti to cis-syn. And this is a reason why the channelrhodopsin adapt to a lower conducting state upon longer illumination. This is necessary for the algae to cope with high light intensity, but this is disadvantageous for the neuroscientists because they don't want this protein to inactivate in continuous light. So you can make some mutants to prevent this, but how the mutation prevents this photoreaction is unknown. We have no explanation for that. So a little more engineering. Probably Carl have spoken about this from his perspective, which is a little different from ours, but synergetic somehow. So let us look at this glutamate 90 of the central gate. We replace this by an arginine. What happens is it flips down from this orientation into this orientation, and the disadvantage is that the central gate is always open, which destabilizes the protein, but it's still closed under normal conditions. The very surprising thing is that the selectivity changed from a sodium channel into a chloride channel by a single mutation, which is a kind of strange. So you see we have chloride influx, but it looks like an outward current because this is a negative charge, so it looks like sodium efflux, what we saw at the beginning, or potassium efflux. And here you see the water distribution in blue, calculated by molecular dynamics, and you see the chloride distribution. And what you see here in the mutant, the pore is a little wider, and the chloride concentration is higher in this protein. So just one mutation changes the system quite completely, and this mutant was applicable, but it was not the final solution because it still had some proton conductance left. So if proton and chloride go into the same direction, then they're inhibiting each other, so as we should call it. And the reversal potential is not identical with the chloride reversal potential. But this was easy to remove by a mutation of this glutamate, first in the entrance, at the extracellular side and at the central gate. And then you had a pure chloride channel, which you can see here from the current voltage relation of the eye clock that we designed, which is at the same place. So the reversal potential, which is this one here, was at the same place as a reversal potential for the GABA-ergot receptor, which is this point here in the current voltage relation. So this looked promising because now we could use this light-activated chloride channels for inhibition. Something is missing here. It doesn't matter. So I like to switch a little bit to another application. A demand from the neuroscientists was to direct the channelrhodopsins in neuronal vesicles to control vesicle filling and vesicle fusion by light. That was difficult. So what we did is, so Franziska Schneider in my group did, she made a construct of channelrhodopsin and arch. Channelrhodopsin as a light-activated ion channel and arch as a light-activated ion pump, and intercalated these proteins into synaptophysin. And in addition, she he interspersed two indicators, MK for expression and fluorine for pH sensing. The advantage of this system is it goes exclusively in the vesicular membrane for reasons I don't understand. We only know that the synaptophysin goes into the vesicles exclusively. No understanding why this is the case. So what we should do it to fill this vesicle up, we should block the proton ATPase with bufalomycin, which is a reagent known for a long time, and then we can replace the filling, or we can control the filling by light. And what happens is, after a while, incubation for half an hour, the postsynaptic current almost completely disappears, and if you switch on the light, it recovers after 40 seconds or so. And you can here see the light periods. The pH drops very rapidly, and then you can see the recovering of the vesicle fusion. So this is a system which can be easily used for real-time studies on vesicle fusion. It has a big disadvantage. It cannot be used in animals, because if you feed bufalomycin to animals that are dead in a few minutes, so it's not so good for your experiment. So the question is, can we change this system into a light-activated ion channel so that we can fill it up and release the protons with the light, which would be a better experiment for in vivo studies. The difficulty was that channelrhodopsins were not expressible in this system, so we had to work with these pumps. So let me go to the pumps. The pumps transport protons along a well-known pathway that has been studied over the past 4 decades. And we know from spectroscopy the sequence of this proton transporting system. And what we need is to convert the pump into an ion channel. And originally, we thought that this would be easy, and it took us some time. Although each amino acid has been mutated in the past, but the spectroscopy was only done in detergent solutions. So the influence on the current voltage relationship and the transport power was unknown, even after 7,000 publications about Bacteriorhodopsin, which is quite a few. Another interesting point is, this is an NMR experiment, and it shows the differences between the M state, intermediate, and the dark state, and the changes that occur in Bacteriorhodopsin are very small. Okay. So we looked at the pathway that releases the proton from the retinal shift base to the extracellular medium, and what happens is you can look at this proton water network, or this amino acid water network that has been studied by infrared spectroscopists like Klaus Gerwert and Joachim Hebel and others. And here you see the outward directed pumping, which is pretty much independent on the pH. When you make a mutation at this arginine, you switch this pumping system into a passive system, which gives you a large outward current at pH 7.5 and 10, and inward current at pH 5. So something had been modified in this system. When you mutated this one, you have large outward current. So what we have done is we changed this amino acids and converted the system in a light-gated proton channel, just by changing a single amino acid. But the system is not ideal, because at 7.5 it has some pumping activity left. So only if you apply a shortened gradient, you shift it from a real pump into a real ion channel. So we convert it into bidirectional proton channel, which is either inward rectifying or outward rectifying, as we call it from our electrophysiology business. To understand this in more detail, Roman Fudim in my group made crystals of this light-driven ion pump, and it's interesting that this light-driven ion pump that we have used here comes from Cocomyxa, an Arctic alga. And the reason is that the photocurrents of this Arctic alga in eukaryotic cells like oocytes or Hex cells are much larger than the photocurrents we can generate with Bacteriodopsin. So we made the crystal structure and solved, we analyzed this crystal and solved the structure, and this was very disappointing at the beginning because the structure was almost 100% identical to the structure of Bacteriodopsin. Although the Bacteriodopsin behaves quite differently in this mutational studies. The only exception is the position of this arginine here is different, and there's an extra arginine, here's a zoom here, which is connecting the proton release groups. So this little change produced some hope that this structure, which took us some time, is useful. And the answer is it was indeed. So what is shown here, he replaced one of the tyrosines here by glutamine and removed the arginine to one side, and then he converted this pump activity into a passive system. And this could be ideally used for neuronal application. Now, I like to make things short. In the next step, after this engineering, we turn to light-activated enzymes, which might also be interesting from an evolutionary or biochemical aspect, and also by an application aspect. So first, we worked on a light-activated or photoactivated cyclase from a soil bacterium, which was discovered by Wolfgang Gärtner and Abalozzi from Parma. And she sent us a clone, and we found that this protein is producing cyclic AMP, and this cyclic AMP, which produced in the light, can be used for opening of secondary cyclic AMP activated channels. The disadvantage of this system is that color tuning is not possible because this is a flavin system. And the second disadvantage is that it shows some dark activity. It shows an activation in the light, but the dark activity is significant. So to use it in optogenetic experiment was difficult. The next system that we studied is a rhodopsin guanylate cyclase, and it's also interesting because this was identified by Suela Gomez from Sao Paulo, and she's a physicist who moved to microbiology about 10 years ago. And she worked on this strange fungus, and she discovered a rhodopsin sequence that was directly connected to a guanylate cyclase sequence, and we expressed it in oocytes and Hex cells. So it indeed works as a light-activated guanylate cyclase, which can be used in optogenetic experiments to activate light-activated sodium or potassium channels with cyclic GMP. So here's one experiment. We expressed it in these large oocytes, so the current can be graded with light intensity. And if you switch off the light, it goes back. And you can do several experiments. And interesting is that the light activity is very large and the dark activity is extremely small. And what you see here at the bottom is not the dark activity of the rhodopsin cyclase. This is a dark activity of the other cyclases that are found in the cells without transfection already. Okay, so this system is very nice for controlling cyclic AMP. So the next thing is to convert it from cyclic GMP production in cyclic AMP production, and for that purpose, Uli Scheib in my lab solved the X-ray structure of this cyclase domain, and certainly we can use it to look into the details and mutate the different residues, and she finally achieved the conversion into a very productive adenylate cyclase with very low dark activity. So now I come to the last part, because here I'm in a biology department. I like to speak a little bit about our more recent experiment, and the difficulty for understanding the photoreceptors is not only to do the biophysics, it's also the biological function, and that was hampered over many years by the lack of reverse genetics in Chlamydomonas. So what we need for better understanding of the natural photoreceptors is we need reverse genetics. Because in our antisense approaches, it's not possible to incorporate mutants because they are also silenced. And the other thing is, if we produce an antisense strain, we lose the strain very rapidly, at least the phenotype. The other thing is that the complexity of the sensory photoreceptors in Chlamydomonas is much larger than we originally expected. So when I started to work in Chlamydomonas, it was believed that they contain a single rhodopsin and a single blue light receptors. And the situation is more complex. So what we purified biochemically are these two proteins called chlamyopsin 1 and chlamyopsin 2. They are encoded by a single gene, which is alternatively spliced. One gene has a retinal binding site, one gene product has a retinal binding site, and the other one doesn't. So from this experiment, we proposed that this is a light gated ion channel that we were looking for, and we have shown ourselves a year later that this is not the case. If you reduce this concentration 20-fold, no change in phototaxis. It was a wrong gene. Next, we studied this Channelrhodopsins, originally termed in the database chlamyopsin 3 and chlamyopsin 4. We know the biophysics very well, so I explained it to you. We also know that the Channelrhodopsin 1 is a dominant channelrhodopsin in most algae, but not in all of them. Some algae have a high amount of Channelrhodopsin 2, but in most algae, the Channelrhodopsin 1 is the most dominant. Another interesting thing is that Channelrhodopsin 1 is regulated under very different conditions. Channelrhodopsin 2 is permanently expressed to the same level. Then we got into a discussion with John Spurridge. He proposed that this is not working as a channel in Chlamydomonas. He proposed that this is connected to transducer molecule in a similar way as in prokaryotes. A sensorodopsin is linked to a transducer, or in E. coli, a chemical receptor is also connected to transducer. And what we found is we found sequences, these are in gray here, 1, 2, 3, 4, 5, 6, 7, 8 transducer molecules. All of them are directly connected to their own rhodopsin, which is strange, but it's also interesting because the rhodopsin is connected to the transducer, then it's connected to a response regulator, and many of them are finally connected to a guanylate cyclase as well. So we have here a two-component system in many cases, which is integrated in a single protein. Okay, the function of these 8 so-called histidine kinase rhodopsins is unknown. We were not able to express this in xenoposoocytes, so we cannot say anything about these proteins unless we have the genetic approaches done. In addition, we have 4 cryptochromes, an animal cryptochrome, a plant cryptochrome, and 2 cryptochrome dashes. The function is also not known. There's a publication from Maria Mittag about animal cryptochrome, and she shows that these are responsible for some protein expressions, but the general function is not known. Then we have a phototropin. I'll come back to this protein as well. And we have a UVRA receptor, UVR8 receptor, which is sensitive for UVB light, and this is known for many plants. This is studied by Roman Ulm in Geneva, but also the connection between phototropin and UVR8 is unknown. So let me focus on the phototropin first. Oh no, first I have to tell you what the complications in Chlamydomonas are. If you establish a DNA recombination system, you have to apply some template which shows homology between your target site and the template, and then you can do recombination and integrate your marker number 1 into the genome. If you have one single crossover, you integrate the whole plasmid, including marker 1 and marker 2, which looks like this. If you have double crossover, you get something which is a proper integration, how I call it, with double crossover. Only marker 1 is integrated. Alternatively, if you apply a plasmid like this, it can align with any homology region in the genome, produce a double crossover, or whatever crossover, and then you get a non-homologous end joining. And the difficulty in Chlamydomonas is that this process is about 10,000 times more likely than the integration by homologous recombination. So what has been demonstrated about 20 years ago by Pete Lefebvre, that you can integrate a selection marker into the genome and select the clone. He showed this for a single crossover with such an integration. In the past 20 years, no one achieved to optimize the system in such a way that you can select or that you can produce a non-selectable gene from Chlamydomonas. So we worked on phototropin at the beginning for several reasons. So phototropin, probably what you know from higher plants, phototropin controls photoorientation of the leaves, opening of the stomatal cells, redistribution of the chloroplast, opening of the leaves, and so on. Phototropin consists of two photoreceptor so-called loft domains, because these loft domains are sensitive for light, oxygen, voltage, two of them connected to a kinase. And then we have a modification of this in some plants called neochromes. They have, in addition, a phytochrome domain linked to it at the N-terminal side. Okay, so we studied the loft domains over years. Other people did as well. This is a structure we sourced from loft 1 of Chlamydomonas. And the essential reaction is a photoreaction between a cysteine here and the 4R position of the flavine chromophore. So you produce a thioadduct, which is fluorescent. So what we did at the beginning is, some years ago, you see this is an experiment from 2009, Boris Sorin, now working for 8 years in algae biotechnology in Israel. So he made an experiment with single-stranded DNA and tried to get a double crossover between loft 1 and loft 2 domain. And this finally worked, so he produced something which is a loft domain connected to a selection marker. And it worked. Surprisingly, we got a phenotype, so this was all not necessary. We could have immediately looked at the colonies, but we didn't know. So we got, let's say, 5 different transformants. This was not functional, so the phototropin was still intact. And here we saw 4 colonies that had this selection marker as integrated. To say the truth, for the project, for this project or for this method did not work in our hands for any other protein, because obviously it is difficult to produce a selection marker which is connected to a domain of the protein that is well enough expressed so that the selection marker is functional. And we never got it to work with a membrane protein. Anyway, the phototropin mutants were interesting. What they regulated is the size of the eye spot, which is shown here, because it was known that in the light, the eye spot size is reduced. It's a kind of adaptation of this algae. And what you also know is that what we discovered then is that the amount of the channelrhodopsins in the eye are also dependent on the light intensity. And this is a mutant which shows large eyes already also at high light intensity, not only at low light intensity. And the rhodopsin concentration remains high under all light conditions. An interesting function of the phototropin. So we can complement this with a total kinase, then this regulation is recovered, or we can complement it with a phototropin that has inactivated loft domains, and then this is not recovered. Okay, so this is complementation. Still large eyes at high light intensity, which is not good for the physiology. Another experiment we did together with a photosynthesis expert from Grenoble, what they showed is for wild type that the so-called high energy quenching of photosystem 2 is blue light dependent, not only photosynthesis dependent. So what they showed is, some years ago already, if you illuminate it for a couple of seconds with high light intensity, then you get fluorescent quenching, and this fluorescent quenching has an action spectrum, which is shown here, which is a large peak in the blue. What they also discovered earlier is that this is mediated by a light harvesting complex in Chlamydomonas called LHCSR3. Only if this light harvesting complex is expressed, then you get high energy quenching. If you deplete this protein, LHCSR3, you get a phenotype which looks like this. This is this RPQ4 mutant. When you remove this phototropin, you also get this phenotype, complete defect of high energy quenching. You see here, phototropin mutant dies at high light intensity in a similar way as a light harvesting complex mutant. And here we complemented this either with a total phototropin or with a kinase alone or with a loftomine. And interesting is when you express a kinase, this one here, it's permanently active. You could expect that the light harvesting complex is also permanently expressed, but this is not the case. So the interesting finding is that you need the activity of the phototropin and the activity of the photosynthesis, and when both activities come together, then you express the light harvesting complex that mediates high energy quenching. Okay. Difficulties with a single-stranded DNA-based recombination. Preparation of pure, long, single-stranded DNA is time-consuming. Integration of selection marker never worked with a membrane protein. And up to this stage, the system only worked in a single flagella deficient strain. We were not able to transfer the systems to any other strain for unknown reasons. Next, probably many of you do gene modification by using nucleases. So the next we try to establish are zinc finger nucleases, which are unspecific nucleases, but you can target them to selective or specific areas of the genome by addition of specific zinc finger domains on both sides of this homo or heterodimeric nuclease. What we did to optimize this system, we integrated the target site of the channelrhodopsin into the selection marker and selected for active zinc finger nucleases. And only if this is repaired here, you get a positive clone, and then you can look whether your channelrhodopsin gene has also been deleted. So it was interesting that this method worked after we optimized it. And what was important is we had to adjust the light-dark cycle. We had to include a heat shock period before transformation. Then we transformed the system and incubated it at elevated temperatures. And then we could select paromomycin resistant mutant. And with this system in hand, you can see you can easily detect the modified system because we integrated a small tag. And we were able to get channelrhodopsin 1 and channelrhodopsin 2 mutants, and also we get mutants for the chlamydomonas 1 and 2, to finally, after 20 years, find out what the function of the chlamydomonas 1 and 2 really is. The difficulty is that not always homologous recombination occurs. You also can sometimes get large integrations of let's say, ampicillin-resistant genes into that cutting site, which makes it difficult to handle. But anyway, these are two mutants where the integration was exactly as expected on both sides. Here we didn't apply a donor, and then something else happens. You get small deletions and small additions by a mechanism we don't understand completely. So we use the Channelrhodopsin deficient mutants to demonstrate the functionality, and I don't want to go into the details. We use certainly a light scattering assay, not this assay from 1866 anymore, to speed it up a little bit. And you can demonstrate that if you plot the activity versus a light intensity, and if you delete the Channelrhodopsin 1, you get a sensitivity reduction of a factor of 1,000. So the mutants are very valuable, and they are stable in contrast to the antisense mutant. Okay, so zinc finger nucleases work well in Chlamydomonas for gene editing, but zinc fingers are difficult to design, are expensive, are time-consuming, and there are only a few good target sites per gene. And this is in Chlamydomonas complicated because any gene is so much interspersed with all kinds of introns that the design of functional zinc finger nucleases is complicated. So then the question certainly arose after this was functional, what else can we do? And the answer is probably not so difficult. We can use a CRISPR-Cas9 system. So what I think is that this person is the one who discovered that system, even if it's not so much even if this person is not so much in focus of the current discussion. Because the current discussion is too much focused on applications, but this, I think, is the person who discovered the system in bacteria, and he published a paper in 2000, Biological Significance of a Family of Regulated Spaced Repeats in the Genome of Archaea Bacteria Mitochondria. Yeah. He reported that these sequences matched SNPs from genomes of bacteriophages, and this finding led him to hypothesize that the CRISPR is an adaptive immune system. I think this is the key finding. Okay, how does it work? We have a Cas9 system. Probably almost all of you learn this in the course. And what happens is you can supply a CRISPR RNA and a tracer RNA, and you can connect them, and then you can supply it to your cells, and then in hope that you can use it for deletion of the gene of interest. So what you need in Chlamydomonas, you don't need a human adapted codon, a human codon adapted Cas9. You need a Chlamydomonas codon adapted system, and certainly you need a gRNA which is connected to the U6, and then you can apply it to your cells. So we only bought this little fragment, incubated with a tracer RNA. So this is a CRISPR RNA, which is bored, incubated with a tracer RNA, incubated with a recombinant Cas9 that we produce in E. coli. And then we can transform the cells. And what we found is this is rapid and efficient to delete Chlamydomonas 1, Acri, and these are the number of clones out of 96 that we tested. So in one case you have 16, and in one case you have only 1. But the interesting finding also was that the homologous recombination of both sides is less efficient than with zinc finger nucleases. So we got many mutants. We used different templates, plasmid, single-stranded RNA, double-stranded RNA, and so on. An interesting finding that we received recently is that we can use wild-type strain, which is less efficient respective transformation, but it's more efficient respective homologous recombination. What happens is strange. Sometimes you have perfect integration like this one here. Sometimes you have a different additional integration, and sometimes you have integration of fragments and pieces like this. So the system is complicated, but my conclusion is after 15 years journey, we are now able to specifically delete and modify, this is not perfect yet, the modification, any gene of interest in the green algae as Chlamydomonas. So this is my group in Berlin. And we are funded by different sources. None of these foundations give money for Chlamydomonas homologous recombination, interestingly, because it took us too much time. And my conclusion is science is like a boat race. So we do boat racing once in a while. you need good material, which means a laboratory which is well equipped. The journey must be fun, the crew must be socially competent and well trained, and the competitors should be more or less equal. Only then the finish is enjoyable. I'd like to mention a few people. These here, this is Irina Sitsova from St. Petersburg, and Andre Greiner, Simon Kelterborn, and our technician Heidi Ebers. They established the homologous recombination during the past 3 years. And at the end, I'd like to mention a few people, or a few colleagues that played with us together. And this is, first of all, Carl Deiseroth. He transferred all the biophysics knowledge from about Channelrhodopsins to the neuroscience. Thomas Oertner and Christine Guy as well. Samuel Noreki, who is a crystallographer in Tokyo. Patrick Scherer is the one who helps us now in Berlin to solve the structures ourselves. We do NMR with colleagues, theory with Markus Eltner. John Kennis is doing femtosecond spectroscopy. Benjamin Rost and Thomas Jens, they do optogenetics in compartments. Reinhard Seifert, he works with us on cyclic nucleotide-gated channels. And Giovanni Finazzi, Grenoble, and June Minigava, they work on higher energy sequencing with that. And in the end, I'd like to mention that I always was pleased to work here in Israel, and I hope that our science, in collaboration between the two countries, are helping our societies, that they bring people together, tear down the fences and walls, and help to save our planet. Thank you very much for your attention.
Fantastic. So, two questions. First one, you mentioned molecular dynamics.
Yeah.
I don't know if you can go back to one of your slides where you show the entire pathway of the sodium potassium putative permeation pathway. And you have a beautiful negatively charged. Yeah, that's a good one.
This one?
Yeah. So you mentioned molecular dynamics, and I was wondering, in fact, using molecular dynamics calculation, one could easily here, relatively easily, I would assume with the Warshall, calculations, etc., you could actually delineate the putative potassium-sodium permeation pathway.
Yes.
I say that because I would assume you have a very unique situation where you have a central gate which you would have a tremendous energy barrier when closed. So I think it's a classical opportunity for delineation with molecular dynamic calculation, this pathway. Have you tried that?
We tried that with Markus Eltner many times, and the result was discouraging. And the reason is the following. What molecular dynamics can do is they easily can calculate the water distribution. What they can also easily do is they can monitor motions of amino acid side chains. What they cannot do is they cannot, unless they use quantum mechanical calculations, they cannot really calculate proton transfer reactions. So they can assume a protonated residue X, and they can do the calculation again. They can use a different protonation state, and then calculate it again. But to predict proton transfer reactions or conformational changes of the structure, which is included or necessary for opening the gate, is difficult to predict.
So you could not face a high energy barrier in the central?
No, the problem is the following. What we do not have is we don't have the structure of the open state. If we would have the structure of the open state with the exact organization of the gate in the dark state and in the light state, then MD simulation could calculate the transition from one state to the other. So what they request from us is that we supply the structure of the open state, and then they say, then we will start our business.
Tough one. So the second question was quite amazing to me. You're finding that when you converted the E90 to an arginine, you actually had a chloride channel which is constitutively open, right? Or
only at the central gate.
Only at the central gate.
Yeah, so the inner gate is closed.
Right.
Still.
You still need the light.
Right. So
Absolutely.
if this would not be the case, this channel would be useless.
So the question is, I'm thinking loudly in terms of evolution. This actually shows that you don't need that many changes in this paradigm protein in which you can convert to all kinds of channelrhodopsins and in converting to all kinds of ions. Could you tell from your tremendous findings here how far can you be, or how many mutations will you need here to convert such a sodium-potassium channelrhodopsin to a chloride, fully functional chloride channel?
So in this case, we made 3 mutations. We made the mutation here, it's a glutamate, and then we mutated this GLU101 and this GLU82
well, the outer, inner, and
to prevent the
central.
Yeah, this was only the case to prevent the residual proton selectivity. So if you change the gate, sodium conductance is completely blocked, but you have some residual proton conductance which can be prevented by doing this incompatibility. So probably Carl reported yesterday, he worked in parallel, not he had a postdoc, Andre Brandt. He provided a proposal to Carl's lab to construct a potassium selective channel. And I know this because this postdoc originally worked as a graduate student in my lab. So his proposal was a potassium channel, and I was very skeptical. I think to produce a high selectivity of potassium over sodium with such a system will probably not work. But he is a very persistent student or postdoc, so what he did is he made hundreds of mutations in Carl's lab. Yeah? In most cases, he didn't even the mutation. He bought hundreds of genes, yeah? Because money is enough available there. Yeah? So, and what he only detected, he detected the shift of the reversal potential to negative values because he directly tested it in neurons. And so 2 parameters. First parameter is the amplitude of the current, and the second parameter is the negative reversal potential. Always in hope to produce a potassium channel. And what did he find? A chloride channel, which had very similar properties as our chloride channel. And you might ask the question, why he didn't simply exchange the glutamate? And the answer is, he used this channelrhodopsin 1,2 hybrid, where this mutation is not possible. For some reasons, if you change this glutamate in C1, C2, what Andre used, then you don't get the chromophore-in anymore. Because the positive charge obviously prevent protonation of the shift base for some sterical reason. So he introduced 10 other mutations, changing the polarity of this ion-conducting pathway, and he ended with a protein which was very similar to the channelrhodopsins that we designed on a biophysical perspective. Very interesting. And then he did this in parallel, and then we teamed up again and optimized both of them together, which was quite nice. So I'm still convinced that the potassium selective channel is much more difficult to produce than a chloride channel. Because a chloride channel is not ion specific. He conducts bromide, iodized, nitrate, and all kinds of stuff. So this is not so difficult to be selective for negative or positive charge, but to be selective for potassium without any sodium conductance is difficult. Yes?
Regarding the genetic engineering method that you tried, I was wondering if you tried using a negative selection marker, it should eliminate most of the other options that are discovered process.
Yeah, it's a very interesting point. But the question is, negative selection markers are not so available or not so easy to handle in Chlamydomonas. Maybe we talk about this. I would like to have one. Yeah, there is a protein a mutation which can inhibit sulfate usage of the cell. I don't forget the name of the gene. Then you have a color indication, which might go into the direction that you mentioned. Okay.
Any more questions? No? So.